In this video, we will discuss about the lipid-rich cells transport lipids to the tumor cells via ABCA8a. So, in the previous experiment, we have understood that there is a transport of the lipids might be occurring from the lipid laden cancer-associated fibroblasts to the tumor cells. So, here they will confirm these things and also find out that it's the ABCA8a gene that helps in the transport of the lipids from the lipid laden cells to the tumor cells. So first they validate that whether the lipids are transported from the lipid laden cells to the tumor cells or not, and for that the plan of the experiment is shown in the panel A, where we can see that what they did is they shot the cancer-associated fibroblast from the tumor cells and label them with this body pi C16 and culture them in the tumor cells for the co-culture them with the tumor cells for the 4 hours and then analyze the tumor cells by flow cytometry. And what they have find out is that there is an increase in the body PIC signals in the KPC1199 compared to the control case. And when they compare this cancer-associated fibroblast from the KC-derived KC-derived tumors and KSC-derived, when they use cancer-associated fibroblast from the KC derived tumors and KSC derived tumors, they find out that in the KSC derived tumors the increase in the body pi C16 signal is high, indicates that lipid transport occurs more in the KSC derived cancer-associated fibroblast compared to the KC derived cancer-associated fibroblast. And when they check this flow cytometry analysis using different combinations of the cancer-associated fibroblast, they find out that cancer-associated fibroblast, when they used from SETD2 knockdown tumor cells, there is a high increase in the body pi C16 signals compared to the case where they use SETD2 wild type or SETD2 wild type plus SETD2C cancer-associated fibroblast derived from these two conditions. Indicates that the lipid-rich transport is more predominantly occurs in the SETD2 knockdown derived cancer-associated fibroblast compared to the other case. And so far they have validated that there is a transport of the lipid occurs from lipid laden cancer-associated fibroblast to the tumor cells. Now they need to find out the mechanism or what are the genes involved in this transport of the lipid. So in order to do that, they study they check the literature and they find out that ABCA8a genes is responsible for the lipid transport from the lipid laden to the other cells. So in order to validate that, what they use is they use the iPSC cells with this body pi C16 signal and culture them in the tumors. And after 4 hours, they check the iPSC and the tumor cells by flow cytometry analysis. And what they have find out is that body pi C16 signals is transferred from iPSC to the KPC1199 or the tumor cells. It can be seen from the column 2, where we can see that the signals goes down for the body pi C16 and transport to the KPC1199, and this occurs when the ABCA8a is overexpressed. And when they did the reverse, that is when they knock down ABCA8a, they do not see an increase in the body pi C16 signals. It can be seen from the 4th column, where we can see that this body pi C16 signals is high in the iPSC, but it is lower in KPC1199, indicating that the this lipid transport does not occurs from the iPSC cells to the KPC1199, and the ABCA8a is responsible for this transport of the lipid from the lipid laden cells to the tumor cells. And now they wanted to check whether the fatty acid transport is occurring from the iPSC or whether the fatty acid transport is occurring from the iPSC or there is some other transport occurs that is controlled by ABCA8a gene. So what they did is, in that case, is that they use the body pi C16 labeled iPSC cells and then take out the culture medium and fraction them in different size, that is 100 kilodalton, 10 kilodalton, and greater than 100 kilodalton, and 10 to 100 kilodalton. These are the 3 condition media that they use, and use those condition media to culture the tumor cells and then analyze the results by flow cytometry. What they have find out is that for the case of this 100 to 10 to 100 kilodalton, they see an increase in the body by C16 signals, indicates that it's the fatty acid in the culture medium that helps in the transport of the lipid, that helps in the transport of the lipid from the lipid rich laden to the tumor cells via ABCA8a transporter or ABCA8a reporter, ABCA8a gene, because this increase in the body by C16 signal is occurs when the ABCA8a is overexpressed in the iPSC compared to the case when ABCA8a is not overexpressed. And in the paper they also said that there is an increase, that is slight increase they have observed for the greater than 100 kilodalton, but in the figure they shows that there is the increase is non-significant. So their focus is mainly on this 10 to 100 kilodalton, and this is also in line with the in vivo conditions that most of the fatty acids are found in this range. Then so far they did their experiments in the ex vivo environment, and now they wanted to check their results in the in vivo studies also. So what they did is they used the pancreatic cancer cells with SETD2 wild type and SETD2 knockdown and with different conditions. And what they have found out is that when the iPSC with so they inculcate not just the pancreatic tumor with the SETD2 wild type SETD2 knockdown, but also the iPSC with the following conditions. So what they have found out is that when the iPSC ABCA8a is overexpressed, then they see the increase in the tumor weight. But when this ABCA8a is silenced, they see the decrease in the tumor weight. And this indicates that ABCA8a has a role towards this lipid-rich transport, and that in turn affect the OXPHOS activation and the tumor proliferations and its growth rate. And these results are quantified on the right hand side, that the tumor weight is increased when the SETD2 wild type is used, and in both the conditions, in almost all the conditions, that is till this ABCA8a is overexpressed. But when ABCA8a is till ABCA8 is overexpressed, but when this SETD2 knockdown cells is used, and in that case what they have found out is that when the ABCA8a is knocked down, they see a significant decrease in the tumor weight. They see a significant decrease in the tumor weight, indicates that ABCA8a has a role towards this tumor growth rate or tumor proliferations. And when they use the orthopedic SETD2 knockdown pancreatic tumor cells with ABCA8a overexpressed and ABCA8a knockdown, and then they find out is that there is a increase in the tumor volume, weight when they use ABCA8a is overexpressed. When the ABCA8 is downregulated, they see the decrease in the tumor volume. And these things are quantified on the right hand side, where we can see that the pancreatic weight goes down when they use ABCA8a knockdown in the tumor cells. Indicates they further substantiate their previous point that ABCA8a has a role towards this lipid-rich transport. And the H&E staining on the panel K also gives similar results. That is, when the ABCA8a is knocked down, the decrease in the lesion area or the cancer affected area. And the quantifications also shows that ABCA8a knockdown shows the decrease in the lesion area. And finally, they use the flow cytometry results to show that ABCA8a is knocked down. They see a decrease in the cancer-associated fibroblast formations, but when the ABCA8a is overexpressed, they see the increase in the cancer-associated fibroblast formations. So this all in combined made a point that lipid-rich cells or lipid-laden cells or lipid-laden cancer-associated fibroblasts transport lipids via ABCA8a transporter to the tumor cells. And if you need to understand more about this results or experiments in detail, you can go through the reference that is provided in the description. So that's all for this video. Thank you for your time.